Okay, this is lecture 2, part 1. We're going to continue talking about blood. Now, the one lineage of cells that I haven't spoken about are platelets, which come from megakaryocytes. So, hemocytoblasts differentiate towards, they proliferate, in other words, they multiply, and differentiate towards a cell called a megakaryocyte. The megakaryocyte stays within the red bone marrow, and little fragments of it bud off or bleb off the surface with some membrane around, and those are the platelets. And platelets are important for stopping breaks in the vascular system. They form what's essentially a temporary plug. Here you see the differentiation pathway, the megakaryocyte differentiation pathway from a hemocytoblast stem cell to megakaryocytes, and you see here the platelets blebbing off of the surface and getting released into the peripheral blood. The platelets are small. These are, if you want to have some idea of the size, there is just one platelet right here. And this is a red blood cell, which is approximately 8 micrometers across. The function of the blood is to distribute substances, regulate, maintaining blood levels of particular molecules that are dissolved in it, and to protect the body. And we're going to go through these. In terms of distribute the blood or transportation, the blood transports oxygen from the lungs to the rest of the body tissues. It picks up oxygen in the lungs, goes back to the heart, and then gets pumped out of the heart, the left side of the heart, to the systemic circulation, going through the arterial side of the circulation, then down into the capillaries where gas exchange can occur. O2 leaves the blood and goes into the tissues, and CO2 diffuses out of the tissues and goes into the blood. It also transports nutrients from the GI tract, picking up nutrient molecules that have been digested in the space of the gastrointestinal tract. The food you eat gets broken down into its building blocks, which for proteins is amino acids, and polysaccharides it's monosaccharides, and lipids it's glycerol and free fatty acids. And not that they contribute a great deal to in terms of nutritional value or nutrients. Some DNA or RNA gets broken down, I suppose, but typically that's ignored in terms of nutrition. Those essential building blocks that are now available for absorption will be absorbed through the GI tract into the bloodstream or into the body. Metabolic waste products are also transported away from the cells to the lungs and the kidneys for elimination. And hormones from endocrine glands are secreted from endocrine glands into the bloodstream, or distributed around the body, and will act on target organs by binding to specific receptors on those target organ cells. In terms of regulation, the body helps to regulate and maintain a balanced temperature, normal pH, and volume, overall volume. You do have some differences in temperature in the body, but essentially overall body temperature is about 37 degrees Celsius. If you get overheated, then there are mechanisms to radiate heat away from the body. And if you're too cold, there are ways of maintaining the core body temperature. In terms of normal pH, body tissues, we've talked about this, body tissues use buffer systems. I did discuss one, which is the carbonic acid bicarbonate buffering system. And in terms of volume, the circulatory system always needs an adequate volume. If you don't have adequate overall volume within that circulatory system, you will not have adequate pressure. And without adequate pressure, you do not get appropriate perfusion of all the parts of the body with blood that's needed. Last is protection. In terms of protective effects of the body, it has its own ability to prevent blood loss and also to prevent infection. Blood loss is prevented by activation of a number of different soluble plasma proteins that are constantly floating around in your plasma in the blood. Most of those, or all of those actually, are synthesized in the liver and secreted into the bloodstream, into the plasma. And also blood loss is prevented by activation of platelets. The initiation of clot formation occurs when a vessel is broken or torn or has some sort of opening in it, in the wall. So what we're talking about is hemostasis, which literally means stopping blood. It's a process that involves a series of several different physiological reactions designed to stop any bleeding. There are three phases that occur in rapid sequence. First, there's characteristically vascular spasms. Immediate vasoconstriction occurs in response to injury. The blood vessels do have, not all of them, but any blood vessel of some reasonable size will have some smooth muscle tissue around the periphery of it, and that will constrict in response to injury, so that reduces blood loss. The second process that occurs is, or phase that occurs, is platelet plug formation. And then the third is coagulation or blood clotting, and we're going to go through each of these. Well, let's start actually. I'm not going to go into any detail about vasoconstriction. I mean, it's simply the contraction of the muscle to narrow the blood vessels so that less blood can seep out. We're going to start with platelets and their activation occurring with a tear in the vasculature. Remember that the entire cardiovascular system is the inner side that's in contact with blood is lined with endothelial cells. It's paved with a single layer of endothelial cells. These cells form a smooth, low resistance surface that allows the cells and the plasma to flow past. But if there's a tear, then what you get is exposure of the connective tissue material that's just below, and it's shown here in blue. What you get is exposure of the connective tissue material just below the layer of endothelial cells. These here are the endothelial cells lining this is a capillary. Now, that connective tissue is basement membrane. It's made up of fibers of connective tissue, collagen fibers, and you can see that some of the collagen fibers now have penetrated into, or it's not a terrific diagram. They're showing the collagen fibers within the bloodstream, but they originated from this layer of connective tissue, the basement membrane here. So what you get is exposure of collagen fibers, and platelets will start to adhere and form a plug when exposed to collagen. So you get platelet plug formation, and that triggers the clotting cascade, in which a series of soluble proteins are sequentially activated and activate each other as they go along from one to the next in the sequence, until finally the last one in the sequence is activated, and it forms insoluble fibers, which form the called fibrin, which forms the actual clot. Now, in terms of platelet plug formation, as I said, damage to the vessel endothelial cells will expose the underlying collagen. The platelets don't actually stick to each other or to the endothelial lining. They adhere to the collagen itself, and they use one of the clotting factors or clotting proteins that I mentioned, that series of soluble clotting factors in the plasma, and it's called factor VIII. Platelets will release some of their contents following adherence, and these are some of the molecules that are involved, that are inside, that get released. And you get more platelets attracted to the site, sticking to the exposed collagen fibers, and with enough of them accumulating and sticking to those fibers, you get platelet plug formation. The actual platelet plug does not keep growing and expanding wider and wider, because it's limited by a molecule called prostaglandin I2. So the platelet plug formation is restricted to the immediate area of the injury. Now, this is a abbreviated diagram showing an abbreviated version of the coagulation or clotting pathway that goes to form those insoluble fibrin fibers that make up the clot. There's a false colored electron micrograph. False colored electron micrograph means a photograph taken with a electron microscope and then colored in. Those photos are always black and white, but it's colored in, and you can see quite nicely red blood cells and the fibrin strands forming that blood clot. So it's quite strong fibers that form from what's originally a soluble molecule. Well, as I said, if we'll start at the top here, as I said, what's happening is that here you see a layer of collagen connective tissue underneath the endothelial cells that line the blood vessel. And with a tear in the blood vessel, you get exposure of the collagen fibers and accumulation of the platelets, so you get a platelet plug formation. Now, platelets will release chemical compounds that make the other surrounding or nearby platelets more adherent. A key component that platelets add to the clotting or coagulation cascade is a molecule on their surface called platelet factor III. It is not just, and I'll explain it in better detail in the next few slides, but it is not just exposure of collagen fibers that starts the cascade, but also tissue factor, a molecule called tissue factor released from damaged tissue cells. So that plus the platelet factor III plus tissue factor plus some calcium and the other soluble clotting factors lead to, and this is now a detailed description of the very last steps in the clotting cascade or coagulation. You get formation of prothrombin activator. Prothrombin activator acts on the inactive prothrombin in the plasma to form thrombin. And thrombin acts on fibrinogen, which is an inactive soluble protein in the plasma, to form insoluble fibrin strands. Now, this is just the last tail end. There are many steps before this, and in each step you get activation. As you can see here, I'll do it in blue here. Activation, activating of a inactive, in this case prothrombin to thrombin, and then activation of an inactive, that means soluble, inactive fibrinogen to form fibrin. What I mean by activation is that these molecules, once activated, have enzymatic activity, and they will act on their substrate. So here, the prothrombin activator is acting as a enzyme and acting on its substrate, which is prothrombin. So it cuts or cleaves prothrombin to form active thrombin. And when I say active thrombin, I mean now this thrombin has enzymatic activity, so it can act on its substrate, which is fibrinogen. And the fibrinogen is activated, cut, cleaved it undergoes a biochemical reaction to form insoluble fibrin. So at least at this point we have prothrombin activator acting on prothrombin to form thrombin, and then fibrinogen activated by thrombin to form fibrin. So the coagulation reaction, in fact, involves transformation of a liquid to a gel that has fibrin strands in it. It's insoluble fibrin strands surrounding lots of red blood cells, and that's what the clot is. There are two major pathways for coagulation to occur. They each have a different activating signal, and initially are separate pathways, but they then converge further down. They are the intrinsic pathway and the extrinsic pathway. The platelet factor 3 molecule that I mentioned earlier on the surface of platelets in the platelet plug promote the final steps in the series of reactions. And the final steps, of course, as I told you before, is prothrombin activator acting on prothrombin to convert it into thrombin, and then thrombin acting on fibrinogen to convert it into the insoluble fibrin mesh. Okay, so this is the electron micrograph I showed you earlier. And this is the slide we just went through. I don't know why I have another copy of it, but let's move on. We're going to go into more detail now about the steps that occur in here. And this slide has much more information, and it's not as difficult as you might first imagine. I can tell you that anyone who's going to take physiology at university, and most health science students have to take physiology, will have to learn it, so might as well learn it now. As I said, there are two pathways, activation pathways for the coagulation event, and one is the intrinsic and the other is extrinsic, and they differ in activation and molecules involved until they merge at this point, activating factor 10 here to 10A. All the factors are shown in Roman numerals, or most of them are shown in Roman numerals, and the active form has a subscript A. So let's start off with the intrinsic pathway. The intrinsic pathway is activated via the exposure of collagen the connective tissue just underlying the endothelial cells. So if there's an injury and there's bleeding, you have to have, pierced through that and expose some collagen. So that initiates the activation. The platelets will cling to that those collagen fibers, and what you get is activation of factor 12 to 12A, which in turn activates 11 to 11A, which in turn activates 9 to 9A. So we're going from 12 to 11 to 9. Live with it. A lot of these things often have historical reasoning behind it, because as they were discovering the different factors, they named them and then discovered another one was involved earlier. So anyhow, 12, that's what often happens. That happens with another kind of type of cascade like this, cascade type of pathway, the complement pathway. Anyhow, it's 12 to 12A, 11 to 11A, 9 to 9A, and 9 also involves 7A and calcium. And then platelet factor 3 on the, or release from the aggregated platelets in the platelet plug will, along with factor 8, form a complex. So you get a 9-8 complex here. So you're going 12, 11, 9, and then with 7 and 8, you get this complex. 7 is involved in complex and platelet factor 3, and that activates 10. So, if you were going to learn it, then I would go 12, 11, 9, 7, and 8 to form 9 and 8, activating 10, all converging on 10. The other pathway, the extrinsic pathway, is activated due to a molecule called tissue factor released from damaged tissue cells. And when you have an injury that causes bleeding, you will have other types of cells that are injured, not just the, and this is non-cellular. The collagen is non-cellular. It's secreted collagen fibers. But here you will have some cells damaged, and they release tissue factor. And so that's another way that the same cascade is, or similar cascade is activated, and you get activation of 7. And then tissue factor with 7 activates 10. So on this side we have 12, 11, 9, then with 7 and 8 combining, you have 9, 8 complex activating 10. And on this side you have tissue factor and 7 activating 10. And 10, when activated, and in this activation pathway, you have contribution from requirement for calcium and platelet factor 3. And 5, you'll get prothrombin activator. And of course, you already know from the previous couple of slides, I showed that prothrombin is activated to thrombin, and then that activates fibrinogen to fibrin. And in fact, although usually they're called, you don't hear the number too often, very rare in fact, prothrombin is 2, and thrombin is 2A, and fibrin is 1, and fibrinogen is 1, and fibrin would be 1A. I should tell you that there is another factor with a higher number, 13. Remember, it all started with 12 here, but 13 is important in that it's involved in tightening up and cross-linking of the fibrin gel, and we might mention that later also. All right. Okay. So this is going over what I talked about, that they're initiated. Pathways are initiated by intrinsic factor. This is collagen fiber triggered. This is tissue factor triggered. So you have a tissue damaging event resulting in some bleeding, and you get a series of protein molecules that are soluble in the plasma. All of them are called procoagulants. And each factor, or and each pathway will cascades towards factor 10. Factor 10 is activated, complexing with these to form prothrombin activator, and then you have the pathway going here and cross-linked here. I want to point out to you that at each of these activations, there's a cleavage of the inactive soluble protein to form the active form. And as I told you before, the active form essentially is a enzyme and enzyme activity. And remember that enzymes, of course, they're all proteins. These protein enzymes, they're catalysts, as you know or should be comfortable with, and they can act on many substrate molecules. They act on one, it gets released as a product, and they'll go act on another. They speed up reactions thousands, tens of thousands, hundreds of thousands of times that the reaction would normally occur without the presence of the catalyst enzyme. Which means that what you get here, for example, is many 12A acting on many 11, and therefore you get a lot of 11 activated here, and then lots of active 11 acts on 9, so you get way more 9, and then it keeps expanding, and it's called a cascade, like a small stream going over and getting bigger and falling as a waterfall, and then going along again and collecting more water from inlets coming along and getting bigger, and so you get a flood rapidly expanding into what's called a activation cascade pathway. So things get rapidly magnified through this, and you get immediately localized lots of insoluble fibrin strands. Prothrombin activator catalyzes the transformation, cleavage activation of prothrombin to thrombin, the enzyme, active enzyme thrombin, right here. And that acts on fibrinogen to form thrombin, activates, causes polymerization of fibrinogen to fibrin. And the fibrin are insoluble strands that form a meshwork and the structural basis of the clot. So the fibrin forms this gel-like trap, and then cross-linking occurs due to activation of factor 13, which strengthens and stabilizes the clot. Okay, then that's blood coagulation or clotting. All right, here you go. Now, there's a process that occurs that's quite important after a clot forms. It's called clot retraction. The fibrin strands will contract and squeeze the serum from the meshwork of fibrin, and you'll get a much tougher clot forming at the site. If you take a blood sample, you can see this happening, clot retraction happening very easily in a glass vial that's used for blood samples. If you take a blood sample in a test tube, a glass test tube that doesn't have any anticoagulant in it, and where the glass is directly exposed to the blood, glass is an excellent activator of coagulation. So a blood sample is drawn and the whole thing looks red. Okay, like that. If you leave it for about, I don't know, say roughly say half an hour in a test tube rack, then what you find when you come back is that the tube will, sorry, if you look at it for immediately within a few minutes, if you took the cork off and tilted this this tube after the blood was drawn into it, you would find that you had a gel-like sausage, essentially, of blood. It would come out as a solid gelatin-like sausage after just a few minutes. If, however, you left it in the tube, then what you find when you come back after half an hour or an hour is that you have now the red blood cells at the bottom, and above that you have serum. And notice I say serum because serum is plasma that is being depleted of clotting proteins, right? A clot formed here, and with time you got clot retraction, and that's a way that's happening. That's a process that's happening in the body to strengthen that blood clot so that it remains strong and prevents any further bleeding. And here I'm showing what would happen if you poured it out after just say give it a minute or two. 5 minutes would be fine. And here I'm showing now what would happen if you poured this tube out. In that case, if you poured out this tube, you would get something quite different. The yellow serum would pour out, and the blood clot would be still quite solid and would come out as a gel if you shook it out of there. But you can easily, it usually sticks in the tube, you can easily pour out this very liquid yellow serum. Okay, now with every injury, a very important part of injury or damage to any tissue is wound repair. And wound repair after a bleeding cut in the vasculature is an important process and is driven by growth factors. The two key growth factors are platelet-derived growth factor, or PDGF, released from platelets. They stimulate the repair of the blood vessel wall. At first you'll get cells called fibroblasts forming, and they'll form a connective tissue patch made of secreted collagen fibers. So you get fibrin, which is really made of collagen. You'll get fibrin formation. It's insoluble fibrin secreted from fibroblasts. That's what you get with any kind of wound. You'll get fibroblasts proliferating at the site and secreting fibrin. And did I say fibrin? I'm sorry, collagen fibers. There's also another growth factor besides PDGF. There's VEGF, which is vascular endothelial growth factor. And what this does is stimulates the endothelial cells to multiply, so they restore that nice paving, smooth lining in the blood vessel. And so you get repair of the initial injury. You also have, and that takes some time, but you also have, soon after the clot's formed, within 2 days of clot formation, you get lysis of the clot. The clot starts to dissolve. And this is a process called fibrinolysis. And also important that the clot should be gradually taken apart and destroyed. And that's a normal physiological response after a clot forms. Are there factors limiting the growth or formation or extension of a clot? You can't have the clot going on and on and on. Yes, there are enzymes that are active and they're, acting on the plasma proteins, but it still has to be maintained or restricted to the site of injury. You can't have your entire blood system, vasculature, the blood in your vasculature clot. That would not be good. So there are two homeostatic mechanisms. This means balanced, maintaining a balance, and I'll talk about it maybe more later. It's an important concept in physiology, homeostasis. You have two mechanisms that will limit clots from getting bigger once they start to initiate. One is The rapid dilution removal of clotting factors, activated clotting factors, being washed away by the blood flowing past. So in an area where you have a tear, you still have blood flowing down, past that tear or area of damage, and it will wash away those activated clotting factors, diluting them so much that that they can't act. There are other negative feedback signals that are inhibiting those activated clotting factors, which are produced or released or present in the endothelium throughout the vasculature. And they include heparin, which is it is used as a drug. Maybe some of you have heard of it being used as a drug administered to people to prevent blood clotting, but it is a natural substance produced by the cells lining the blood vessels. There's another one, antithrombin 3, and protein C, which is also very important. So these anticoagulant molecules and the active action of the blood flowing past diluting restricts the clot to the area of injury. Unnecessary clotting, abnormally unnecessary, unneeded, inappropriate, let's call it, clotting is prevented by the structural and molecular characteristics of the endothelial cells lining the blood vessels. They provide quite a smooth, non-resistive anticoagulant surface. The adhesion of platelets is prevented by those smooth cells lining the blood vessels. There's also production of heparin, as I said earlier, and prostaglandin, which I mentioned earlier. Sorry, not prostaglandin, prostaglandin I2. It's very important, I2. There are many different prostaglandins. There is also another potent anticoagulant present, that's vitamin E. Now let's talk about disorders, but I think at this point it wouldn't be bad. I can keep going a few more slides. Let's talk about hemostasis disorders. Sometimes a clot will develop and persist in an unbroken blood vessel. So that's an inappropriate formation of a clot. That is called a thrombus. So a clot forming in an unbroken blood vessel is called a thrombus. And here you see a thrombus forming along the wall of this blood vessel right here, in this area here. There's that clot forming there, and that's a problem. Because just like a boulder in a stream, it causes turbulent flow, which enhances the possibility or increases the possibility of further extension of the thrombus, allowing it to get bigger, because you have turbulent flow instead of laminar flow, which you have here in a normal unbroken flow through a normal healthy blood vessel. Now, a thrombus can cause serious damage. It could block circulation, so downstream you get inadequate O2 to the tissues downstream. You get tissue hypoxia, which means low oxygen in those tissues, inadequate supply of oxygen to the tissues, and it's downstream of the thrombus. In fact, you can get death if you get complete occlusion and no blood flowing, or completely inadequate flow of blood. You can get death of the tissue that's downstream of that blood vessel. That is called an infarct. Local death of tissue due to tissue hypoxia because of inadequate blood supply. It is always a wedge-shaped area of dead tissue. Infarcts are wedge-shaped. The reason for that is that if you have a blood vessel supplying a certain area, it's going to split into, smaller blood vessels as it goes along. I'm not drawing this very well, but you get the idea. It's splitting into smaller and smaller blood vessels. And so what happens is if you get a blockage, you're going to get a wedge-shaped, characteristically a wedge-shaped, and by wedge it means like a three-dimensional kind of structure blocking an area of tissue. It would be like a pyramid type shape downstream, right? It would, I'll put it here from the blockage onwards, it would be like that, spreading down, quite far down actually, right? Now in the heart, if that occurs in the heart, if you get a thrombus formation in a coronary part of the coronary circulation, the arteries in the blood or arterial in the blood, if you get a thrombus in there, you will get an infarct, and in this case, because it's in the heart, all these words have, you know, they have clear meaning. You'll get a myocardial infarct. So this is tissue death downstream of a thrombus within the myocardium, the muscle of the heart. And that's basically the real term for a heart attack, a myocardial infarct, or MI. Now, another serious problem with thrombus formation is that you can have a thromboembolic event. You can get a thrombus, which is you can get an embolus, which is a thrombus that breaks away and floats freely in the bloodstream. Here is a thrombus that is formed in a vein. I know it's a vein because there are these valve structures, and only veins have those kind of valves. Arteries don't. And not surprisingly, actually, it's forming just behind that valve leaflet. The flow is in this direction, shown here. Yeah, flows in that direction. And, you know, just behind there you might have some more, especially if it slows down too much or gets stopped because the person is not moving, you know, they're sitting for too long or stay in one position for too long, or they get a clot forming, a thrombus forming, and it breaks off. When it breaks off, it's an embolus. Now that embolus will travel through the bloodstream until it hits a smaller, narrower blood vessel. So this embolus in a vein can go back to the heart, get pumped up to the lungs, and get stuck in a small blood vessel in the lung. And that's a problem. So pulmonary emboli do sometimes occur. They impair the ability of that area in the body and the lungs to take up oxygen. Sometimes it goes up to the brain. It gets pumped out of the heart, and depending on where it's formed in the circulation, how far it gets, it could go up to the brain. Then you get a cerebral embolus, or emboli. Emboli is plural for embolus, single, and that is what is called a stroke. But it's also it's essentially a cerebral infarct. It causes cerebral infarct. Infarct is a good word to learn. So that's not good. How do you prevent that? By using therapeutically pharmaceutical agents to prevent undesirable clotting. Aspirin has effects that inhibit this molecule that comes from, if you recall, from platelets, that contributes towards clotting. Heparin has anticoagulant activity, reduces causes prolonged clotting time. Warfarin also can be used. It is a drug which commercially is called, when used in humans as a drug, is called Coumadin, but the real term for it is warfarin. Also happens to be used as a rat poison often. People buy Coumadin laced food that rats like and then lay it down, and the rats eat it and end up bleeding to death, because we're always bleeding all the time. We're getting little bleeds. It's a constant battle to prevent any serious blood loss or inappropriate blood loss, and platelets and the coagulation cascade all prevent that. But sometimes they're too active, and people need to be treated with anticoagulant therapy to prevent inappropriate, undesirable clot formation. Are there certain characteristic disorders of hemostasis? Yes, and then I'll describe them to you. One is called DIC, or disseminated intravascular coagulation. For you guys, DIC is good. It's easier. I teach this because it covers, you have to think through the process. It's not just memorizing things, and it's, it should make sense to you what I described based on what you've heard in the previous slides. This is due, or this causes, this is due to widespread, minute, but multiple widespread blood clots forming throughout the blood vasculature. If you get many tiny blood clots forming, what happens is that you get a depletion of the clotting factors, the proteins, soluble proteins involved in the clotting cascade. And so now the person, after they had disseminated, which means widespread, intravascular, which means inside the intact blood vessels, right? Coagulation events, tiny little blood clots forming. Their blood cannot, they're fine, but their blood can't clot anymore, and if they do have any tear anywhere, they will get bleeding. This is a very, very potentially fatal complication in pregnancy right after delivery. It can occur for other reasons. It can be associated with septicemia, which is presence of growing microorganisms, bacteria in the bloodstream. They could trigger it. Or an incompatible blood transfusion where the blood is not matched properly. In terms of pregnancy, when a woman delivers, the baby is delivered and then the placenta separates off of the wall of the uterus. The placenta is the area where the baby circulation and the mother's circulation were closely, not in direct contact, but very closely in apposition to each other, and so that's where gas and nutrients and metabolic wastes are exchanged, and that separates away from the wall. If during delivery you get amniotic fluid getting into the mother's circulation, which can occur, it can trigger widespread tiny little blood clots, disseminated intravascular coagulation. And now she has a major site in the uterus where the placenta was, that is still, fairly raw and could bleed. And these kind of people can exsanguinate. They can lose all their blood within minutes. It's quite dramatic and terrible. Thrombocytopenia there are many causes of thrombocytopenia. It is a low or deficient number of circulating platelets. People who have thrombocytopenia, low platelets, have these small purple blotches in their skin because they're getting little bleeds all over. And you can see it on the surface of their skin. They get these little purple blotches. They're called petechiae. Spontaneous widespread hemorrhaging. Everyone, normal people, have spontaneous widespread hemorrhaging. But it immediately gets stopped, mostly by platelet plug formation, but also if it's slightly bigger, also probably some coagulation. Thrombocytopenia can be caused by a number of different things: destruction of the bone marrow, suppression of the bone marrow. When a person's treated with radiation or certain drugs, you can get bone marrow suppression, so there's not enough platelets being produced. If there's a tumor of the bloodstream, the white cells, for example, like leukemia, you get it, the bone marrow being taken over by that type of leukemic cell, not enough megakaryocytes there to produce platelets. Thrombocytopenia, I'm not 100% sure about this number because things change. I teach the physiology of it, not diagnosis or treatment in any detail, because it'll not be up to date. But in general, platelet count less than 50,000 per cubic millimeter is indicative or diagnostic for thrombocytopenia. Now, another disorder of hemostasis are bleeding disorders because of deficient synthesis of the clotting proteins that are involved in the clotting cascade. The procoagulant proteins, all the different clotting factors. I told you they're synthesized in the liver and secreted into the bloodstream. Now, people who have deficient levels of these procoagulant proteins or clotting factors can get very severe bleeding events. It could be the cause of that could be due to vitamin K deficiency, because vitamin K is required for proper normal coagulation or clotting and formation of the clotting proteins, or inflammatory process in the liver so that it's not producing the proteins at appropriate levels. Or another very commonly seen type of deficiency is due to cirrhosis of the liver. This is damage to the liver due to chronic injury, often seen in kind of thing is seen in alcoholics, because when you drink alcohol, you do get injury to your liver. If you take in more alcohol, you will have injury that's seen in the liver, and the liver can repair itself very quickly. But alcoholics will do that every day or often enough and damage their liver continuously. And with liver injury, tissue injury in the liver, you get fibroblasts proliferating. Remember, I told you with the wound repair, you have fibroblasts proliferating and secreting collagen, connective tissue, fibers. So in a person who's chronically damaging their liver and injuring their liver, they get the liver is taken over with lots and lots of connective tissue rather than the liver cells that should be there, and so they have deficient production of clotting factors or procoagulants and often bleed. A genetic deficiency of a clotting factor is possible also. And people who have that kind of genetic deficiency are hemophilias. Are hemophiliacs. They have hemophilia. There are many types of hemophilia depending upon which clotting factor protein gene is abnormal. The most common type of hemophilia is factor VIII or von Willebrand factor deficiency. That's called also called von Willebrand's disease. But that protein has been, the gene has been cloned and is produced commercially in labs, and that, those kind of people are easily treated with recombinant factor VIII. So with hemophilia, people with bleeding disorders or not just hemophilia, other bleeding types of bleeding disorders, you can get any bleeding can be prolonged, painful, often in the joints, because the joints are the most common area of small bleeds. All of you, as I as I said to you earlier, you get spontaneous bleeds occurring all the time, but they're immediately stopped. The joints are a typical place where you get bleeding in hemophiliacs. Treatment is by transfusion of fresh fluorescent plasma, injection of the missing factor if the missing factor is available because has been produced as a recombinant protein. So there are treatments, not for all of them, but many types of many possible treatments. Okay, the next part of the lecture is going to be on blood transfusions. We're going to stop here. This is the end of Lecture 2, Part 1.